From talking CMC impurities in this workshop, we shift back to the non-clinical arena and share thoughts on how to develop these drugs from a safety perspective. These are my disclosures. And in this talk, I will focus on and discuss platform considerations to bring these drugs to IND states and beyond. Some safety testing will obviously be done early in the process during lead optimizations to screen out bad actors, but the talk will primarily focus on the characterization of the toxicity and DMPK profile to support clinical development. Very much an open door, but there won't be a one recipe fits all strategy, primarily because of the diversity in approaches and oligo designs that we have today. There may be unique concerns with some mechanism of action and less with another, or with some design features or chemistries that require de-risking. So you need to tailor it to the specifics of your entity. And how you deliver the drug to patients impacts your clinical strategy as well. So local injections into the brain or eye will not result in the same potential risks as when given systemically through subcutaneous IV. And the fourth element being the therapeutic indication, which has impact on the benefit-risk equation. And although safety testing should always be adequate, there may be justifications for abbreviated programs that hold up in the context of a rare and fatal disease, but not for a more common disease. As an extreme example, you may appreciate non-clinical package is much leaner. In the case of Millicen, a true N of 1 drug for a single patient, compared to inclisiran, which is now in clinical phase 3 testing and may be used by millions of people. So bottom line, all of these factors impact how to design the non-clinical testing strategy, and you need to tailor it to the specifics of your oligo and the use of your oligo, and then align with regulators on your plan, whereby you carefully justify deviations from the expected. When it comes to expectations by the regulators, ONDs are regulated as chemical entities. So the principles of small molecule guidelines apply. Particularly ICH-M3 is one of the key papers. But they also possess properties resembling biologicals, which are regulated by ICH-6. Things about their larger size, higher target selectivity, predictive organ distribution, and degradation pathways. So elements of biological drug development should also be taken into account, and a hybrid M3S6 approach is therefore a sensible departure point. No OND specific ICH guideline, but recently Japan has issued a preclinical safety guidance for oligos, and a reference in the back, so that will be helpful. And on an ongoing basis, the OSWG publishes papers providing scientific advice on a number of relevant safety and CMC topics. Now, the classical way to look at toxicity is that division between related to pharmacology, not related to pharmacology. And you can apply that same paradigm to oligos, and we get that, you know, this is a very familiar classification whereby most of the oligos toxicities are chemistry related. So, you know, nicely lining up with, you know, the properties of small molecule. Most small molecule toxicities are pharmacology related. This classification into pillars nicely lines up with two major advantages of ONDs over small molecules when it comes to safety testing. And one of that is the rational design element related to the hybridization, and the other one is the predictive class profile related to the chemistry-driven effect. So the rational design can facilitate the evaluation of hybridization-related toxicities, particularly off-target effects. So you can de-risk by in silico screening, using tools and identify other complementary RNA sequences in the human genome, which can be followed up in a human cell model using gene expression analysis methods. And if needed, detailed assessments of these off-target genes to assess whether the hazard actually translates to a certain risk. For de-risking of on-target effects, there should be secret homology in the targets of one of the animal species used in safety testing. If not, but the target is present in the animal species, then a surrogate sequence could be used. However, carefully assess the need for that, thorough evaluation and prediction of loss or gain of function effects. Because you could rationalize perhaps why knockdown or restoration of a particular protein function would not be detrimental. In that case, you could justify the absence of a pharmacological response in species on paper. The other advantage comes from the predictive class profile, which helps to bring focus in the safety assessments. And the major driver for the class profile behavior are the physical chemical properties that come with the modified chemistries of that phosphate ribose backbone. So phosphorothioates, for example, large polyanionic molecules readily bind to plasma proteins, which give them a different PK and tox profile than the neutral backbone morpholinos, for example. And class profile behavior is well established for known chemistries, like phosphorothioates with 2-MeO-2-O-methyl modifications, and perhaps some of the bridged ribose modifications like LNAs or CATs are getting there as well. But we should be aware that within each chemical, group, there could be bad sequence outliers that we need to catch early. And with further expansion of the oligo field, there will be increasing nuances to the class profile concept, with new toxicities that may come from novel designs or chemistries, or from successful delivery, getting higher cellular concentrations than before, of increased use of local administration routes, where so-called poor uptake organs like brain and eye that did not see much oligo before now suddenly have massive oligo concentrations, and we need to characterize what that will do to those organs. This slide just shows the established profile, class fingerprint of systemic effects. Won't go into much detail if you have time. Important to note that although it's rare to see effects outside of this fingerprint, we should know that the risk is not the same for each and every candidate in that chemical class, and should be assessed on a case by case. And we have touched on the topic that the future will perhaps bring more nuances to this. There's also remarkable consistency between different sequences in terms of plasma and tissue PK and overall distribution characteristics on this slide. Again, not going into detail, but I like to stress that a plasma exposure is generally not a good reflection of the tissue exposure in these drugs. Concentrations in the tissues that take up oligos is much higher for a longer period of time due to the slow elimination from the tissue. Thus, for assessment of PK-PD or PK-tox effect relationships, kinetics in tissues is more informative than kinetics in plasma. And as a comment related to that, if you directly administer the oligo into tissues like the brain, the eye, or the lung, concentrations will be high in those organs for a long time, but systemic exposure will be low, sometimes negligible, and this minimizes concern of any systemic toxicity. Now let's look back more to the how-to question, how to do the toxicology testing. And general toxicity studies form the central core of the safety package. Major objectives being to determine a safe dose, the NOAEL, as well as the maximum tolerated dose, and that can help to guide the clinical dose range. Also identify target organs to help with the monitoring strategy in the clinic, and estimate safety margins and assess reversibility of findings. Now, I will discuss 4 critical design elements where oligo-specific thoughts may have impacts. So first is the dose range that you want to use in those toxicity studies. And usually you do some dose rating studies as a toxicologist. But in the case of oligos, the advances you can build on previous experience. And also important to note is that most of the oligotoxicities is late onset, so not well reflected in short-term dose ranging studies. Then second element is the duration of the repeat dose studies, and that's dictated by ICH-M3. And for chronic treatment in patients, this should be a 6-month study in rodents and a 9-month study in non-rodents. However, EMA appears to be okay with 6 months rodent studies, and I have experience where 6 months works well for the ophthalmology division of the FDA, whereas other divisions of FDA expect to see 9 months studies. Also, long recovery periods are usually needed because it takes time for tissue levels to drop below thresholds of toxicities. And then a third element is the administration routes that you need to tailor to the intended clinical routes. But you could, if it's complex routes, you could think about mixed programs with the local routes in the non-rodent species and the parental routes, sub-Q IV in rodent species. And as a last element, frequency of dosing in animals should be similar or more frequent than planned in the clinical program. Target tissue PK/PD considerations are a critical driver for determining the dosing frequency. And to bring patients rapidly to steady states, you may want to use a loading regimen in the clinic. Either more frequent dosings in the first period, or initially higher dose levels, and then maintaining it later on with lower levels, with lower doses. But this then, if you do that, that should be reflected or adequately covered in the design of your animal studies. And then the last element is selection of the species. So ICH-M3 demands 2 species for your safety evaluation, 1 rodent species and 1 non-rodent species. And this may be the right thing to do for most programs. However, there may be situations where using only 1 species is justified. And there's an option that is open for biologics in ICH-6. Key, of course, is that the potential risks are adequately assessed in that single animal species. And this may be the case, for example, if 1 species is a truly well-established good predictor of the human responses, whereas other species or rodents are have their downsides. That an example of that is ocular indications for oligos, where the primate eye is a true good model for the human eye. And another example is high medical need indications in ultra rare diseases, with the extreme example of Milacen that was supported by a rat-only program. So in other cases, there may be good justifications for mixed programs where subchronic studies are run in 2 species, and only the the chronic study is run in primates only. So that is a strategy that was successfully used for nusinersen. And then a final comment here that with regard to species selection, you should be aware that there exists species sensitivities for oligos in responses. You need to be aware of that because that may impact your risk assessments. Yeah. I would like to end with 3 slides that list all preclinical activities and highlights in the comment box where there may be opportunity to provide justification to deviate from the default standards. Of course, always in the context of your specific drug, your specific therapeutic indication and patient population, but supported by argumentation from the class profile or by published data. And especially the latter is important. So I stress to all of us to get more safety data out in the public domain to help and support regulators in their guidance to the oligo programs. So this first slide lists, studies that, generally kept it in an IND enabling package. And then studies that are usually conducted beyond IND or even, post-marketing potentially, if you align with the regulators on that. And then a series of studies in the DMPK field. Most important comment on these 3 slides is actually in the subtitle, to make sure that the safety package for your specific candidate meets regulatory expectations. You need to work with the regulators and seek scientific advice on your programs where needed to get their buy-in early and to be able to adopt their suggestions in your program. And with that, I'd like to end this talk. Hopefully have given you insights that help in the design of your next non-clinical testing strategy. And thanks for your attention. I'm happy to take any questions.